What are the, so read the question from start to finish.
The sites where crossing over occurs are referred to as
And the potential answers are?
stoma, teres, was it chiasmata?
Chiasmata?
You say that's piasmata?
Chiasmata.
Oh, okay.
Yeah, like the Greek letter chi.
Chiasmata.
Chiasmata. And that is going to be the correct answer. Chiasmata.
What?
If you guess. Tetrads are the structures. But
Shut
this
up.
is
It says it
asking.
in the back of the book. It says something about in the back of the book about
Yeah, but
crossovers.
it's
wrong.
Yes, but it's asking for the actual site of crossover. This is called a tetrad. And this is the structure. Okay? This is a structure. And the structure is called a tetrad. But it's not asking for the structure. It's asking for the site. The site of crossovers.
Yeah.
I'm glad you asked that.
Is chiasmata.
Okay, so can we just like start from the beginning then?
If you would like, yeah. Let's go.
Alright, the cells of a whitefish are undergoing mitosis. Whitefish have 80 chromosomes. How many sister chromatids would there be in prophase of mitosis in a whitefish?
Would it be 180, 160?
I did
Okay,
40.
so is it asking for pairs of sister chromatids or sister chromatids?
Sister chromatids.
chromatids.
Okay, so remember, with sister chromatids, you start with 80 sister chromatids. But you duplicate those during S phase. During S phase, we duplicate.
Maybe 160.
And it would give you 160 sister chromatids. Because you're duplicating them. Remember, your final cells are going to wind up with the original number of chromosomes. So these are our chromosomes, and we want to know how many sister chromatids those are going to produce. There's going to be 160. Okay? Next. What's the next question?
A whitefish cell that contains 80 chromosomes would be classified as.
Okay, so we start with 80 chromosomes. So if we perform mitosis, how many chromosomes should be in our final cells if we start with 80? 80. That's what denotes them as 2N, which would also be classified as a
Is diploid?
diploid. Good, okay. Now if these cells performed meiosis, how many?
1N, and it'll be
40.
Oh.
And they would be classified as
Haploid.
haploid.
Close. I was close. Okay.
So it would be D.
So what's the question again?
A whitefish cell that contains 80 chromosomes would be classified as A haploid, B diploid, C having DNA content of 1N, and then D is A and C.
Okay. Well, they're diploid and they are technically 2N. So if there's no 2N on there, the only correct answer is
Or diploid.
diploid, right?
Correct.
Correct.
Correct. Okay. All right. Next.
I really can't tell because I put it out in black and white. So I want to say because where's the book?
Find the picture in the book.
I want to say this one is.
Hey, I got it. Don't worry.
Oh, you got it?
Yeah.
Okay, he's got it in color.
He was already ready.
Okay.
So it's
Metaphysics.
Let's see. Where are those? I think those
What
are
did he
the
have?
other center. Right.
Because mine is
Here.
black and white, but I
so
answered
it
is
looks like
metaphysics.
man.
Yeah,
It's
I just
exactly
printed
that,
mine
so
out.
it's going to be metaphysics.
Yeah, metaphase.
Wow. So I guessed it right.
Which structure has been separated during meiosis 1?
Can you tell us the question number?
7.
That's number 7.
Number 7. So what structure is separated in meiosis 1? So remember, homologous chromosomes, right? Remember, meiosis 1 separates homologous chromosomes. Meiosis 2 separates sister chromatids. Okay?
So it's
So if it's asking for meiosis 1,
right.
the structure that's being separated is going to be homologous chromosomes.
And will we need to know the difference between chromosomes, chromatids, and then
heard, right? Chromatin is just the DNA wrapped around protein. Chromosomes are when you're not performing division. Okay? It's at the end in telophase or at the very beginning in interphase. Chromatids are what they exist as from prophase all the way through anaphase. So when they're in that duplicated structure that has that butterfly shape, that's going to be a pair of sister chromatids, and you're going to call them chromatids. Once we've divided them and they're no longer stuck together, then you're dealing with chromosomes.
Oh, okay.
Okay? Make sense?
Totally.
Good.
So
Okay.
meiosis II separates sister chromatids then? Okay.
Meiosis II separates sister chromatids. Meiosis I separates homologous chromosomes.
Number 5, a dog that had the genotype capital B, capital N, and a dog that had the genotype lowercase b, lowercase n, have puppies. Puppies that have the genotype capital B, lowercase b, capital N, lowercase n, have black coats and have poor eyesight but can still see. Which of the following describes the relationship between the alleles that encode eyesight?
That's too much for a little dog.
That's too much of one question.
Eyesight. So the eyesight is poor. Okay, so first off, capital B is for the black coat. Capital N is good eyesight. Lowercase b is for a brown coat. Lowercase n is poor eyesight. Okay. Now you'll notice capital B, lowercase b gives us black for their coat color. So, in terms of that, are we looking at complete dominance or incomplete dominance when we're talking about coat color?
I thought it was
Incomplete.
For coat color, first off, remember, with complete dominance for coat color, and then we'll actually get to the question. But for coat color, we're seeing black and brown mixed, but we're getting black out. That's complete dominance, right? Because one of the two alleles is acting as the dominant allele. Now the question is asking about eyesight. With the eyesight, you get a heterozygote, but you get poor, well, they can still kind of see, but they can't see well. So is it truly the big N's genotype or phenotype that we're seeing? No. But is it technically also this phenotype that we're seeing? No. Poor eyesight here, technically I guess they're blind, right? These dogs are not blind, but they can't see well. So it's a combination of the two, which is why we call this incomplete dominant.
Wow. Bill was right.
Yep.
Said it the whole time.
Yep. So incomplete dominance.
So-
Understand, with incomplete dominance, you're going to see a combination of the two phenotypes. These dogs can see well, these dogs are blind, but the puppies can't see well but aren't technically blind either. Yeah. Me too.
Yeah, like if I take my glasses off, I don't know anything on there.
Really?
Yeah,
Same.
like I have really bad eyes.
Same.
It's crazy.
So that's incomplete.
I got bad eyes, but I got contacts. I'm like, you losers.
I have contacts. My eyes
broke
usually
yesterday
dry
playing volleyball.
All right. Next question.
My mother has brown eyes, big B, small b.
Okay. So hang on.
What number is this?
3.
Mother, big B, small b.
And the father has blue eyes that are 2 small b's.
Okay.
If they have children, what are the likelihood that one of the children that they have will have blue eyes and will be a female?
Okay. So we're going to do a Punnett Square. And we want to know the percent chance that they'll have blue eyes and be a female. So big B, little b, little b, little b. Now, there's a couple of things we can get from this. First of all, we know that the big B is dominant because the mother has brown eyes, while the father has blue eyes.
Would the mom have to have a big B?
And she does.
I'm
Yes.
saying If it were the girl, it would come out have a big B.
To have brown eyes, yes, but if we're asking about blue eyes, just has to have these.
Okay.
Okay? So here, real quick, genotypes that we get, we're going to get big B, and little b, little b. Do we get any big B, big B's?
No.
No, so that's 0%. These other two are going to be 50% or 1/2 for each, right? Because 1, 2, 1, 2. Okay? Phenotypes. Brown versus blue. What are my percent chances of getting each of those? 50% and 50%, or 1/2. Okay, now the question asked the percent chance or the likelihood that you would have a child with blue eyes and a child that was female?
Yeah.
Okay, so are we going to add or multiply? Multiply, right? Okay, what's the percent chance or the fraction that I get blue eyes? 1/2. And means I'm going to multiply. And what's the percent chance or the fraction that I'll get a female? 1/2, right? Which then means 1/2 times 1/2 is 1/4. And if I write that as a percentage, it's going to be 25%.
So they would have to be small b to get female?
No. Remember, female and male come down to the X and Y chromosomes.
Awesome.
Anytime you have a female, that female is always 2 X chromosomes. Anytime you have a male,
Oh, yeah.
that male is always an X and a Y. The mom will always give the child an X chromosome. It's the father who determines if they get an X or a Y. And because there are just 2 that we're dealing with, the likelihood of getting an X is 1/2, the likelihood of getting a Y is 1/2, and that's where we get that 1/2 from.
Okay.
Okay? All right, any other questions?
I don't know. Just look at
Number 9,
Number 9, okay, what's the question, Hope?
it says a single culture of cells is divided into 2 separate flats.
Okay,
One of the cultures is grown in the presence of growth factors,
okay,
and one is not. After the cells have been incubated for a period of time, which of the 2 flats will have more cells in it? Then question number, answer number 1 says the culture without growth factors will have more cells. B says the culture with growth factors will have more cells. C says both cultures will have the same amount of cells. D says it is impossible to determine from the information provided. E says none of the above are correct. So I
Okay.
answered B. Sorry.
so which one did you say would have more?
I said that the culture with growth factors will have more cells.
I'd say you're on the right track, Hope. Remember, this is taken straight from the book. There's a picture of cells being pulled up. This is where we were talking about the things that affect cells dividing. And there is a picture in the book. I believe it's a couple of pages after it shows you the cellular division, but it literally shows this experiment. What it shows is the cells without growth factors have no growth, but the cells with growth factors start spreading across the plate. Okay, what page is that on, Phil?
That's on 133.
133. So page 133 is where they show that experiment to the T. So these are the ones you would see grow. Those you would not. Okay?
The growth factors, and then they'll have more cells.
Yes. As their name implies, these are factors that promote cellular growth. So if we add them to the cells, those cells are going to want to divide. If the cells don't have those added, they're not. They're going to be stuck in that state of G0.
Yeah.
Okay?
And then mitosis occurs in the blank cells, while meiosis occurs in the blank cells.
Somatic and reproductive.
Yep. Somatic and reproductive. Excellent.
What do you have for number 12?
She was right.
What do you have for number 12?
Number 12, I don't even know how to look that one yet.
What's the question?
It says if a father is dead,
Okay.
then lowercase d, 2 of them.
Okay, so the father is deaf, and then they have 2 children?
No, they have 3. The wife is not, no.
Okay, so the
The
mother's
father
not,
is deaf, then lowercase d, but his wife can hear.
okay,
They have 3 children. 2 of the children can hear fine, but 1 of them is deaf. What is the genotype of the mother? And then question A is DD.
okay.
Uppercase,
So when we take the mother's, which is unknown, and we cross it with the father's, we get 3 children. This child can hear, this child can hear, this child is deaf, and the mother can hear. What is the genotype of the child who is deaf?
DD.
Little d, right? Because that was what the father was, to be deaf. So, then the question is, what does the mother have to be so we could get a child like this? Now, the mother can hear, so we know that she has to have 1 big D.
and
Right,
small
So
d.
we don't know about the
Small
other.
d.
Yes, yes,
Okay.
okay?
Oh, okay, I have a question.
Okay.
Which of the following occurs in meiosis but not in mitosis?
Okay,
I think it's homologous chromosomes crossover. Is that true?
I would say that's correct. What are the other options though?
No, all of them.
There are
It's
2
all of the
rounds
above.
of division.
Okay, does mitosis have 1 or 2 rounds?
2.
2. Look at the chart we just made.
Wait. Yeah, meiosis has 2.
Is it number 11?
So meiosis
Yeah.
has 2 rounds,
It's
but
all of the
mitosis
above.
only has 1.
It's this.
Homologous chromosomes cross over, and then what's the last option? Or the last options, I forget.
The cells form an end of, no.
Classified as
It's formed an
1 in, okay.
Oh, man.
So all of those are differences between meiosis and mitosis.
So all of the
Yes,
above
I
have
did.
Yes. Yes, that is correct. You have to be careful about those.
Phil, you are not smart. Why did you pick B? I told him.
Oh, you're
No,
such a
no,
liar.
no.
Jeez, such a liar. Oh my goodness.
Okay, can we go over the true and false really fast?
Sure.
The centrosome.
False. No,
So,
It's true.
the centrosome is a sticky region as the center of a pair of sister chromosomes.
that is false.
Centrosome.
It is the centromere that's the sticky region. Remember, I told you that's where you need to know.
Centrosomes make the spindle thing, right?
Right. This is the centromere.
So smart.
This is a centromere, and if I look at a pair of sister chromatids, this is a centromere.
The centrosome is on top, right?
But remember, some means body, and the centrosome is going to be at the heart of the spindle structure that has all the microtubules that attach at the kinetochore by the centromere. So this is a centrosome. That's why it's so important to know the vocabulary. Because you have centromere, you have centrosome. Okay?
Confusing.
So that's false.
Blood types are caused by codominant alleles.
True.
True. We got
Yeah,
looked at that.
Just
A sperm would be classified as haploid if it
False.
has DNA content of 2 in.
False. Remember the flow
Wait, how's
charts.
that not true?
Because it's one
It's
in.
false.
Remember the flow chart. Look at the flow chart. Okay, is it a reproductive cell? And there were 2 things underneath reproductive cells as well. It was egg and sperm.
uh-oh.
If those are egg and sperm, those are the things that are produced. It's meiosis that you're talking about. And if you're talking about meiosis, it's 1 in and it's haploid, right?
Yeah.
Oh.
Yes. Yes.
This one,
Number
19.
20 is true.
Humans have 22 pairs of autosomes. Oh, that's not right. And 1 pair of sex chromosomes.
It's true.
That is true.
See?
Remember, we have 46 chromosomes total. We have 22 pairs of autosomes, giving us 44. And then we have 1 pair of sex chromosomes. Giving us the additional 2 that adds up to 46.
Okay, matching.
Okay?
Homozygous.
It describes an individual with 2 of the same
Allele.
allele.
Yes. So homozygous. So again, another place we have to be careful because we have homologous and we have homozygous. Homologous means the two chromosomes have the same genetic locations, okay? Homozygous means we've got two of the same allele.
The chromatin is structure composed of DNA and protein.
Yes, that is correct. DNA and protein is chromatin. And it makes up the chromosomes, which are duplicated into pairs of sister chromatids.
Alright.
The chromatin is
It's not, these are all.
Not at all.